Hello, my name is Hyun Yuk. I have a research lab in the Bionanoscience department at TU Delft. As a student, I studied physics and math, but today I spend all my time thinking about the living cell. If you are a student who likes math, physics, and other disciplines that use quantitative methods, then in this lecture I want to tell you why you might fall in love with biology as well. You can quantitatively study the living cell in the Bachelor's Nanobiology program at the Erasmus University Medical Center and at TU Delft. In this lecture, I will tell you about genetic circuits, one of the key topics that you will learn in the nanobiology program. So genetic circuits are molecular circuits that control all cells' behaviors. Before we look at genetic circuits, here is some familiar types of a control circuit. On the left, you see we have an electrical circuit. It has wires that transport electrons from a voltage source like a battery to electronic devices like a light bulb or the central processing unit of a computer. And our society is built on devices that are controlled by electrical circuits. If you open your smartphone or computer, you will find a circuit board that looks like this. It has billions of little wires and electrical switches that process digital information. Just like these circuits, all living organisms like us need to process information. Decisions such as what to eat for lunch and whether or not to continue watching this lecture all depend on cells in our body processing information. But how do they do it? The answer is that cells use genetic circuits. Genetic circuits don't have any wires. They reside in all cells, like in these E. coli bacteria that you see here, and in this gray circle. Genetic circuits look like this. Instead of wires, the genetic circuit in a cell is made of genes located on DNA, which are drawn as these boxes, and proteins that interact with the DNA to turn on and off these different genes. These arrows represent interactions between proteins and genes on the DNA. Let's break this complex-looking circuit into simpler versions. All genetic circuits have three basic elements. They have a DNA. On this DNA, there's a gene. Let's call it gene A, drawn as a box sitting on this DNA. A gene is a DNA sequence that instructs the cell how to make a particular protein, in this case, a protein A. You can think of a gene as a written recipe for a specific protein. A molecular machine, let's call it a slider protein, which lands on this box and then reads this recipe as it slides over it. This slider reads the recipe and makes the protein. Next to the gene is the promoter. It is a sequence of DNA that attracts the slider protein to the gene. By controlling its attractiveness to the slider, the promoter controls how frequently the slider slides through the gene and produce the protein A. So the promoter is as a molecular dial. Like Lego blocks, you can place any type of a promoter next to a gene. For example, a promoter can produce a gene at a constant rate. This means that the amount of protein in a cell is constant over time. A strong promoter makes a high amount of protein A in the cell. A weak promoter makes a low amount of protein A in the cell. Here's another type of promoter. A promoter can be a special sequence of DNA that this green protein recognizes and then sticks to. This green protein is called an activator. It binds to this promoter. It then turns on the production of protein A by recruiting the sliding protein to come here and through this gene. This creates a genetic switch. It acts like a light switch. When there is low amount of an activator, cell cannot make protein A, but when there is a sufficiently large amount of the activator in the cell, then the cell switches on the production of protein A. On the other hand, you can also have a promoter that recruits another type of a protein, like this green protein with an X sign. This protein is called a repressor. It sticks to this promoter and prevents the sliding protein from producing the protein A. The repressor does this, for example, by physically blocking the slider from going through the gene. It acts like a roadblock. This creates another type of a genetic switch. When there are few repressors in the cell, then the cell is making a lot of protein A. But when the amount of repressor is high, the cell turns off protein A. Now let's look at more complex genetic circuits. In this circuit, gene A makes protein A. This protein A binds to the promoter of B and turns on the production of protein B. Here, we see that if the cell makes a lot of protein A, then there will be a lot of protein B as well, due to more protein A causing production of more protein B. So this allows the cell to always do two tasks, one controlled by A and the other controlled by B at the same time. But the cell cannot do just A or just B without doing the other task. Here's another example. Gene A makes protein A that binds to promoter of gene B to lower the production rate of protein B. Here, we see that turning on production of protein A causes the cell to turn off the production of protein B. Therefore, the cell chooses to do either one task controlled by A or another task controlled by B, but never both at the same time. Now consider this circuit. Can you deduce how this circuit works? Can you tell me what increasing the production of protein A will do to the abundance of protein B? Well, this is where our intuition fails. Here, there are three interlocked regulation arrows. To answer our questions, we need to put some numbers onto these arrows that tell us how strongly each arrow influences a gene. This is where we need math. We need equations that describe the changing production levels of proteins A and B, how to derive and analyze these kinds of equations, and then seeing how they match up in experiments in cells is what you will learn in the nanobiology program. An equation called a differential equation looks like this. If you study nanobiology, you will learn how to design genetic circuits, analyze them using these types of circuit equations, and how to see the production levels of proteins changing right in front of your eyes in living cells. Well, that's it. Now you know what genetic circuits are. You also learned how a cell makes basic decisions using simple genetic circuits. Thanks for watching.